going to discuss about bacterial spores. So before discussing bacterial spores, we will look for sporulation stage in this diagram. Clear? So this is showing structure of bacterial spores. This is bacterial spore structure, and these are the steps of sporulation. Okay, so the first step is axial filament formation. This is your DNA. This is forming axial filament. The next step is septum formation. You are clearly looking here, septum is formed. The next step is engulfment of 4 spore. This is known as 4 spore. Okay, this is 4 spore which is engulfed inside this. So this is the engulfment of 4 spore. Is third step. The fourth step is cortex formation. Cortex is formed. The fifth step is coat synthesis. Exosporium is there. It is here. The sixth step is maturation. Okay, and seventh is this release of the spore. Clear? So this is diagrammatic representation. Now we will move for theoretical part. These are highly resistant structures. Okay, and it is formed in unfavorable condition when there is a depletion of exogenous nutrients. Clear? Now bacterial spores are formed within the parental cells, which are called as endospores. So if bacterial spores are formed within parental cells, that bacterial spore is known as endospores, and the remaining part of the bacterium is known as sporangium. The remaining part of the bacterium is known as sporangium. Clear? Now coming to the structure of the bacterial spore. So bacterial spore comprises of several layers from innermost. We will move from innermost to outermost. The layers are, the innermost is core, then you will find cortex, then you will find coat, and the most outermost is your exosporium, as you have seen in diagram. So first will be your core, then cortex, then coat, and then exosporium. Clear? Now the core is the innermost part which contain DNA material. Okay, so core is important because it contains DNA material, and it is walled off from the cortex by an inner membrane and the germ cell wall. Okay, so between core and cortex there will be inner membrane and germ cell wall. No problem here. Okay, now cortex and coat layers lie external to the core. Cortex and coat are externally present to the core and are separated from each other by an outer membrane, and the outermost layer is known as exosporium. So this is the general structure of the bacterial spores. Now we will look for a sporulation, as we have discussed the steps of sporulation, but now theoretically we will look for. So the first stage for sporulation was the axial filament formation. Okay, bacterial cell division occurs and an axial filament of nuclear material is formed, as you have seen in diagram. The second one is your septum formation. Cell membrane is folded inwards to enclose part of DNA and to produce the 4 spore septum. The septum formation, that is also known as 4 spore septum. The third step is, we have discussed, that is engulfment of that 4 spore. Okay, the membrane continues to grow and engulf the immature spore in second membrane. Okay, the fourth is your stage 4. In stage 4, cortex formation was there. The cortex synthesis occurs in the, and where cortex synthesis is occurring, so cortex synthesis occurs in the space between the two membranes. Okay, with the deposition of calcium and DP cholinic acid mainly. Okay, now coming to the fifth step. So fifth step is the formation of coat. This is a protein coat and formation of exosporium around the cortex. This is the step 5. 6 is just maturation of a spore. Maturation of a spore means it has acquired properties of heat resistant and refractility. Clear? Now the 7th is finally lytic enzymes destroy the sporangium to release the spore release. So first is your axial filament formation, then septum formation, then your engulfment of that septum, that is 4 spore septum, then cortex formation, then your coat and exosporium formation, then maturation of a spore, and then release of a spore. So this is the basic steps of sporulation. Now discussing some important point about sporulation. So first important that it is not a method of reproduction. Mind it, sporulation is not a method of reproduction. It is not a method of reproduction because the bacteria do not divide during sporulation. The bacteria is not dividing during sporulation. Clear? Okay, sporulation commences only when growth ceases due to lack of nutrients, and it is a complex process which takes almost 10 hours. Okay, so this is about sporulation. Now we will move for next, that is germination. The next topic we are going to discuss is germination. So it is the transformation of dormant spores into active vegetative cells. So it is the transformation. Germination is just the transformation of dormant spores into active vegetative cells when grown in nutrient-rich medium. Okay, and it consists of 3 stages. Germination comprises of 3 stages. The first is your activation. It is a reversible process that prepares spores for germination and usually results from treatment like heating. So first we will activate that spore. The second is your itself germination. It is the process of breaking of spores dormant state, characterized by there are several characteristics, that is spore swelling will be there. Okay, then rupture of the spore coat, there will be rupture of spore coat, then there will be loss of resistance to heat and other stresses, there will be loss of refractility, okay, release of spore components and increase in metabolic activity. So this is under the germination. And then the last step, that is outgrowth. The spore protoplast emerges from the remains of the spore coat and develops into an active bacterium. So germination, just conversion of dormant spores into active vegetative cells. 3 stages: first activation, then germination. In this we have break spores dormant state, and in outgrowth the new active bacterium has arise out of the spore. Clear? Now we will look for shape and positions of spores. Okay, for a given species, the precise position, shape and relative size of those spores are constant for a given species. These all are constant. Okay, now first coming to the position of the spore. So spores may be central, or spores may be subterminal or terminal. So these are the positions of the spore. Coming to the shape, so thus they may be oval or spherical in nature. Then width, so the diameter of a spore may be same or less than the width of bacteria. Okay, if the diameter of a spore may be same or less than the width of bacteria, or may be wider than the bacillary body, producing a distension or bulge in the cell. That is, so first, if the width of the diameter of a spore may be same or less than the width of bacteria, then it is known as non-bulging spore. If diameter is lesser than the bacteria, in compare with the bacteria, that spore is known as non-bulging spore. Clear? And its example is bacillus. Bacillus has non-bulging type of spore. Which type of spore? Non-bulging type of spore. The second one is bulging spore, in which it is the spore is wider than the bacillary body, okay, and creating a bulge in the cell. So that is spore known as bulging spore, and that is found in Clostridium. So Clostridium has bulging spore, whereas Bacillus had non-bulging spore. Bacillus has non-bulging and Clostridium had bulging spore. So this is about spore shape and position. Now we will discuss some points about sporicidal agents. So spores are resistant to most of the routinely used disinfectants. Only limited sterilization methods are available to kill spores, which we will discuss in our next video, that will be sterilization. We are not going to go for the detail here. Okay, now we will move for demonstration of spores. So demonstration of spore is first by simple, that is gram staining method. By using gram staining method, you can demonstrate spores. Spores appear as unstained refractile bodies in the cells. In gram staining, it will be appear as unstained refractile bodies within the cells. Clear? In gram staining. Now the second method to demonstrate is just modified Zehel-Nielsen staining. Modified Zehel-Nielsen staining. In this, spores are, as you know, spores are weakly acid fast and appear red color when Zn staining is performed using 0.25% sulfuric acid as decolorizer. Clear? Now the third is your some special techniques for endospore, which include the Schaefer Fulton stain, or you can also use Moeller stain. So these two are special technique, Schaefer Fulton stain and Moeller stain, which are used for demonstration of spores. Now next point, that is applications. Clear? Now spores of certain bacteria are employed as indicators for proper sterilization. So it is used as indicators of sterilization. When we will discuss sterilization, then you will get more idea about this. Okay, so they are the indicators of sterilization. Absence of the spores after autoclaving or processing in hot air oven indicates proper sterilization. Some examples: so spores of geobacillus stereothermophilus, spores of geobacillus stereothermophilus, okay, are used as sterilization control in autoclave. So in autoclave we use this geobacillus stereothermophilus as a indicator. Clear? The next, spores of bacillus atrophius. Spores of bacillus atrophius are used as sterilization control in hot air oven. So when we look for autoclave, then you will use geobacillus stereothermophilus, okay, and where you will go for hot air oven, then you will look for bacillus atrophius, clear, as a sterilization indicator. So this is all about your bacterial spores. So thank you for watching.